Okay, welcome everybody. To those of you that are in our Zoom room, ready to discuss with our panel, and to those of you watching us on YouTube, this is day 2 of Next Generation Electrochemistry 2021. And we're going to have another set of events going on at 10 a.m., 1 p.m., and 3 p.m. Chicago time. The panel that we're going to have now is a panel on neuroelectrochemistry, and it's going to be run the same way as we ran the panels yesterday. Not all the events are like that, so stay tuned because this will change as we progress in the day. The goal is, again, having a spontaneous discussion that will be driven by the panelists who are leading experts in the field at hand, in the field of neuroelectrochemistry. They will all give an opening statement. And the idea here is that they'll present some fundamental questions that they view as important in the topic that we're discussing. Always keeping an eye on advances that have to do with electrochemistry per se. And so our goal is that those of you that are watching this, you should start connecting dots, even if the topics seem very different, because you're going to see very common aspects popping up constantly. We had that yesterday and continue to happen today. We have a group of about 40 students and postdocs in the Zoom call. And as yesterday, they will have the opportunity to take the floor and ask questions directly to the panelists so that they can interact directly with them. There's also a live stream. There's a chat in the live stream. If you want to post questions there, we're monitoring that too, and we can always read them. We're going to run for 90 minutes. And like yesterday, Zoom doesn't kick us out, but I want to be respectful of everybody's time. So we'll cut the discussion at 90 minutes unless it's very vigorous and people insist that they want to stay for a little longer. For our panelists, my charge to you is that we want to really hear about what you view as the end of our current knowledge in your domain. What are you interested in learning about? What do you think that the younger generations, when they take over, will have to worry about in terms of research topics? We want to spend most of our time discussing that. It is okay to be controversial. We had some really mild controversy going back and forth yesterday, and that is great because it sparks very vigorous discussion. This is your opportunity to share your thoughts and influence the field beyond your group, so don't be shy about it. We really want to tell the students and the postdocs that are in this call how to approach problems, to think critically. We talked a lot about emphasizing asking the right questions yesterday, and that's a great addition to the specific topics that we may discuss. Attendees, you've seen this already multiple times, so I'm going to go very quickly through it. Get involved, mute until you're called to the floor. If you have a question, make yourselves known, and I'll push you to the floor to ask directly. If you have an issue with your mic or with your camera, don't turn the camera on if you don't have a camera. But if you have an issue with your mic, let me know when you are ready to ask the question or when you want to be put in the queue so that I can read it for you. I love that some of you challenged the faculty in your statements yesterday. That is great. That's what we want. Think broadly, even if this is, like yesterday, not quite the topic that you're working on. Make connections between what you do and what you're hearing about. And you're going to see that there are questions that just keep popping up. With that, I am going to leave the floor, and I'm going to start with the opening statements. So we agreed on an order before we started. And first up is going to be Christy Haynes from the University of Minnesota. Christy, whenever you want, the floor is yours.
Great, thanks. Let's just make sure that you all are seeing slides as expected. I assume that looks okay, Jordy?
Yeah, that's good.
Yeah, awesome. Okay, so I'll start by, of course, thanking Jordy for inviting me, and I'll just say to all the students that are on the call this morning, I think you're super lucky. You've got great panelists here. I'm happy to be here with Jill and Janine and May. And so I'm Christy Haynes. I'm a professor at the University of Minnesota in the Department of Chemistry. I've been here for 16 years now. And I'll just start off by saying that even though this is neuroelectrochemistry, I am definitely the outlier among this crew of panelists. I'm definitely interested in neuroelectrochemistry, but that's not really where my group has been focused. So I think there'll still be a lot to learn by thinking about systems that I look at that are similar to neuronal systems. And I'll also just give you the caveat that actually I was not originally trained as an electrochemist. So my PhD is actually in surface science and nanoscale materials. I didn't really do any electrochemistry until I did my postdoc. And that was with Mark Whiteman at UNC Chapel Hill, which when Jill presents, you will hear that, you know, she is core Whiteman trained as well. So we definitely have a lot in common. So I'll start with a little bit of background. Maybe I can figure out how to not have this floating panel show so that hopefully you guys don't see gray boxes on your screen. So my group's electrochemistry focus has largely been on doing single cell measurements, single biological cell measurements. And I mentioned, you know, I did a little bit of work on neuronal cells back during my postdoc, looking at neurons relevant, optical neurons, for example. But really, my group at the University of Minnesota has focused instead on two types of biological cells. So mast cells, which are critical immune system cells, and platelets, which technically are not even cells because they're anuclear, but we'll call them anuclear cells just for simplicity. And because we're doing single cell measurements, of course, that means we need small electrodes because we want the electrodes to fit onto a cell. So the electrodes we work with, you can see them pictured up here in this scanning electron micrograph. This is an electrode I learned to make in the Whiteman lab, and it's called out as a carbon fiber microelectrode. If you look closely at this micrograph, hopefully what you can see is that the center portion looks different than the exterior. So that's the carbon fiber in the middle with a glass insulating layer. And this electrode has been beveled at an angle so that it can kind of sit flat on top of a single biological cell. The diameter of these electrodes is right around 7 microns. People have definitely worked to make these smaller, and I know you'll hear about some nanoelectrodes in a couple panelists. And you can see the general idea here is simply that we can put the electrode down on a single cell. We can put a small pipette nearby that can essentially initiate a biological event that we want to measure. And specifically, the event we want to measure is exocytosis. So this is the way that biological cells deliver chemical messengers, one way that they deliver chemical messengers to the exterior of the cell. So the idea is that you have a cell, this is a platelet. You'll notice no nucleus, like I mentioned. You can see the cell membrane very clearly, and you can see all these kind of small circles in there. Those are different kinds of chemical messenger-filled granules. So essentially, when the cell is stimulated, those granules move to the membrane exterior, the membranes of the granule and the outer cell membrane fuse, and the chemical messengers are released outside. That's the exocytosis process. And we essentially want to detect those chemical messengers as soon as they're released to the exterior of the cell. So the two major methods that my group uses to do these measurements, you can see them listed here, are fast scan cyclic voltammetry and amperometry. And fast scan cyclic voltammetry is exactly what it sounds like. You're essentially scanning, the potential to see where the redox active species are, and you do it very quickly. And this ends up being important when you're studying processes like exocytosis, because of course that's a biologically fast event. Microseconds, milliseconds are kind of the relevant timescales. So you can't do, HPLC separations and detections where it takes minutes, right? So fast scan cyclic voltammetry is important if you want to identify what a cell is releasing. But once you know what's being released from a cell, often the identity of those chemical messengers don't change in a given cell type. Then you can essentially hold the potential exactly where you need it to be, and then do an amperometric measurement. So in that case, you're holding the potential at one place and you're measuring current. And what you can see down here on the bottom of my slide is one example amperometric trace that's been measured from a single platelet. So we put the electrode on top of one tiny platelet, stimulated it, and then in the stimulation was just the small amount of time you see this little bar. And you can see these beautiful skinny current transients. And each one of these skinny little spikes is one granule worth of, in this case, serotonin that's being released from the platelet. So my group is interested in this technique, both because we want to know fundamentals about how cells store and then release chemical messengers. And of course, FastScan CV tells us what's being released, right? And amperometry tells us a lot about how it's being released. We do a lot of work where we manipulate the cells or the conditions around the cells so that we can understand more about the granule contents. But my group is also really interested in this because we think it gives us really critical insight into various disease contexts. So imagine you have some normal, healthy mast cells, for example, and then you have a set that have been exposed to some exogenous, potentially toxic species, or a set of mast cells from a kind of disease condition. By doing a direct comparison where you get all of this really nice biophysical information, you can understand how the disease or that exogenous toxic species impacts the cells. So my group started out thinking specifically about the toxicity of nanomaterials. So we were using mast cells, which are a critical immune system cell, and then allowing them to be exposed to engineered nanoparticles, which I'd be happy to talk about if anyone's interested, but is definitely outside the scope of this little panel. And we looked at how the nanoparticles disrupted chemical messenger delivery. And it helped us understand a lot about how you would design nanoparticles to avoid disruption. We also are very interested in diseases. So my group has worked specifically with cells from sickle cell disease, platelet syndrome known as Hermansky-Pudlak syndrome, and most recently we've been focused on malaria. And we're also interested in using this method to understand how drugs might affect cells. So you can imagine if you have malaria and you're given a treatment and it's supposed to be targeting the parasite, which is actually in your red blood cells, that means the drug is also near the platelets that are so critical in your bloodstream. And so we're interested in looking at kind of off-target effects of drugs. And so we've worked hard to understand how malaria treatment drugs impact platelet chemical messenger secretion, for example. So that tells you a little bit about kind of where my group's focus is. And like I said, you don't see any neurons here, so I'm sorry for that if you came for the neurons, but I know that these techniques are definitely all relevant if neuronal cells are more your thing. So Jordy also prompted us to think about kind of, there we go, challenges and limitations that we saw in the field. And I think that Janine, is our fourth panelist, is going to go big on some of these things. So you'll see that my challenges and limitations are a little bit more practical more than anything. So a common issue when people are doing single cell measurements include the fact that, of course, we can only detect things that are redox active within the biological, like aqueous environment, right? So things like serotonin, dopamine, octopamine, histamine, those things are all detectable, but there are lots of things, lots of chemical messenger cells released that are not electroactive in the window that we would love to have them be electroactive in. So I think the question, you get used to the beautiful, insightful, real-time biophysical information you get about chemical messenger release with single-cell electrochemistry, and then you can't do it for things like GABA or glutamate, things that people really care about. There has been a lot of effort to adapt these electrodes to make them work for species that aren't typically electroactive in the right window. So people have definitely created enzyme layers on top of these electrodes that have a mediator so that when GABA or glutamate come in and bind, you see production of hydrogen peroxide, which of course you can detect. My group has been interested in taking on this challenge actually by working on a different electrochemical sensing platform, specifically using field effect transistors, although we're nowhere close to the same kind of time or spatial resolution that we have with carbon fiber electrodes. You'll notice in the amperometric trace down on the bottom of this screen that we have these beautiful skinny current transients every time one of these granules fuses with the cell membrane and releases contents. So those transient or burst events are really nice to detect. You can do some gorgeous quantitative analysis, right? I can tell you exactly how many molecules came out of that granule. But a lot of biological processes don't happen in this kind of burst way, right? They happen as kind of tonic, low-level release. And those are much harder, at least to my mind, to quantify using techniques like this. Everybody always wants the most sensitive electrode they can get, because of course that means you can detect fewer molecules. However, there is this balance that we struggle with, at least in the way my group does electrochemistry, where if you have an electrode that has a high affinity for a species, which means it's going to be very sensitive, it also means the electrode is prone to fouling. And I'll just mention that in the types of measurements I showed you on the previous slide, with these electrodes, we can make measurements from about 5 cells, and then we have to swap out the electrode, and we have to repolish it and clean it and recondition it before we can use it again. So there's definitely this hard balance between affinity and fouling. Single cell measurements, I would love to be able to do these in moving fluid. My group has spent a lot of graduate student hours trying to embed these electrodes in microfluidic devices, either so that we could better simulate blood flow, right? We're interested in platelets, or because we wanted to measure from a lot of single cells, one right after another, to build up a bigger data set. And I will just say, in general, what we found is that working with these small electrodes and the small kinds of currents that we measure, we've had a very hard time doing it in moving fluid. The noise that comes from simply having a biological motion is an impediment. And then the last two are kind of bigger challenges for anybody that's doing kind of any type of single cell analysis, whether it's electrochemical or super resolution microscopy or anything else, which is, how do you draw ensemble measurements from single cell data? I love single cell data. I think it's really important. I think biological heterogeneity is definitely critical to all biological systems. However, the question is, how many single cell measurements do you have to make before you actually have the whole collection that describes the biological ensemble? And then the last one, I know Jill is going to tell us some answers for how to do this, but in general, one challenge is, how do you ever make sure that when you're doing these in vitro measurements, so I have platelets sitting on a cover slip in a petri dish that I've isolated from an animal or human blood, or I have mast cells that are sitting on top of some fibroblasts in a petri dish. How do you ever draw big biological in vivo relevant conclusions in that case? So I think it's really tough. It's not just a numbers game. It's also the fact that, of course, in biology, there are many interacting systems. And so just because I see statistical difference in my in vitro data, how does that contribute to biological significance in an in vivo system? So those are the challenges and limitations I thought of. And with that, I'll stop and pass it to our next panelist.
All right. Thank you, Christy. So May Shen is second. May joins us from the University of Illinois at Urbana-Champaign. May, the floor is yours whenever you're ready.
Okay, so good morning, everybody. And thank you, Jody, for organizing this and on the invitation. And it is truly my honor to be here. So I have to say, all of the panelists are really pretty inspiring. Ever since I was a graduate student, I go to the conferences, I have been meeting them. So currently I am assistant professor from University of Illinois at Urbana-Champaign. And before then, I did my PhD study at University of Texas at Austin with Alan J. Bard. So Janine, that's how I actually met Janine in the conference. So, and later on, I did my postdoc at Pittsburgh with Shigeru Amamiya, working on nano pipette electrochemistry and nano resolution imaging for nanostructures, and I'm going to share some of those later on. And thanks, Christy, for a very nice introduction about carbon fiber electrode, exocytosis, as well as amperometry. So to start with, so I would like to give a bit background for neurotransmitter detection. As you all heard the talk by Christy, where like dopamine, serotonin, these are electrochemically redox active neurotransmitters. They can be detected on the carbon electrode based on the oxidation. And electrochemically speaking, there is another group of neurotransmitters. So that is non-redox active. So Christy gave you two examples, GABA and glutamate. Another example here is acetylcholine. So basically all of these non-redox active species, they cannot be detected directly or carbon electrode based on the oxidation. also Christy has mentioned enzymatic modification of carbon electrodes have been carried out for their detection. So far, those still yet to make them to be like a nanometer scale. So which is the focus of the interest in my lab, because we want to probe synaptic neurotransmission. So our strategy for the non-redox active neurotransmitter is by developing the liquid ejection electrodes, which is the interface between two immiscible electrolyte solutions. So how this works is we have a pipette. In our lab, we make them to be nano scale, like nano pipette. And inside of the pipette, you can fill it with one phase, and outside can be another phase. So in the case of our application to detect neurotransmitter, so this outside phase 2 is the cellular medium, and inside the pipette is phase 1, which is the organic solution. So this organic solution inside the pipette and the cellular medium, they are not immiscible. So when we put them together, so then you have an interface between two immiscible electrolyte solutions. So this is the electrode surface for the situation of liquid ejection electrodes. And when we polarize this interface, the current will flow. So this is how we use ITIES electrodes to sense, to quantify, and to detect neurotransmitters. Now, I would like to share a bit about the thermodynamic basis of chemical sensing with ITIES electrodes. Many of you probably have taken electrochemistry class and knows how to derive Nernst equation for the redox reaction from the electrochemical potential. Likewise, the same thing you can do here. So this is my lecture notes this spring semester for a graduate course, electrochemical methods. So we have ionic species I that is at equilibrium between two phases. One of them is aqueous phase, and the other one is the organic phase. At equilibrium, just like redox reaction, we can write down this expression for the electrochemical potential. So that we can expand this electrochemical potential. Then we have this. So this is standard chemical potential. Here's activity term. This is the inner potential. So then we can rearrange this equation, put the inner potential on the left side. Then on the right side, we have this. So then bear in mind this standard Gibbs free energy of transfer equals to like this. So then eventually we can rearrange this to have expression like this. So this is the transfer potential, just like the detection potential for redox reaction. And here is the standard transfer potential. And down here is the activity term. So the way how we can picture this activity term is, so AI water. So this is for the transfer from the oil phase to the aqueous phase. So this is our like reactant, and this is the product. So this is the theoretical basis for the quantitative sensing of chemical species using ITIES electrodes. So today, I'm just going to focus on neurotransmitter detection, but overall, this is a very broad field and has, for the chemical sensing with ITIES electrodes, it has like 40 years history, and it has been used for sensing many different kinds of chemical species, like heavy metals from the environment, protein, things like that. So for the quantification, because we want to quantify the neurotransmitters, because when disease, and disease state, so typically the concentration of the neurotransmitters are much less. So this is an equation we use and has been developed by LiFeU, so where we can get the concentration information from this limiting current. This applies to both nano and micro electrode. So here I'm going to share with you some experimental data from my lab, where you can see for 3 different neurotransmitters, acetylcholine, tryptamine, and serotonin will have very different detection potentials. And here is example from our results to show that we can quantify these neurotransmitters as well using cyclic voltammetry as well as amperometry. And amperometry here we can achieve the temporal resolution of millisecond, which is pretty good. And we have ongoing work in our lab trying to decrease this further to reach microsecond resolution. Okay, so now I can share with you the strategy our lab have developed to detect like cholinergic transmitters, which is one type of non-redox active neurotransmitters. So next thing I'm going to share a bit about how we achieve a high spatial temporal resolution. So on the left side are the nanoelectrodes our lab has been making. There is also several other groups also has been working with nano electrodes as well. And on the right side is a scanning electrochemical microscopy platform. And Professor Janine here is also the expert in this area. And she has written a book chapter before about scanning electrochemical microscopy in the cellular studies. And how this works, basically, we have a three-dimensional nano positioner that you can attach your electrode there. Then after that, you can perform the three-dimensional imaging while collecting chemical information at the same time. So overall, this is a very broad discipline by itself. And we have a whole book on this area and also a whole conference on this as well. And Professor Janine, me, and to our colleagues will organize this conference next year. Okay, so here is some examples of nanometer resolution imaging that have been accomplished with nanoelectrodes and scanning electrochemical microscopy. So on the left side is the nano-resolved images of a single nanopore. Under these blue boxes here is the nanopore membrane that was taken using TEM under vacuum. And on the right side here, this is a result I got when I was a postdoc in the Amamiya lab. So where we used nano ITIES pipette electrodes and scanning electrochemical microscopy to image the ionic flux across this single nanopore. As you can see, we have achieved nano resolution. And here is another example of nano-resolution imaging using SESAM. This is by the group of Michael Merkin to study the catalytic activities of single nanoparticle. So there is also many other emerging area of research in the field of nanoelectrochemistry. So another example is to study a nanobubble by the group of Harry White at University of Utah. So now, I'm going to touch some powerful application of nano ITIES electrodes. So one area of my research is to probe single cell signaling of cholinergic transmitters. So what we bring new to the field is we position this cholinergic transmitter detection nanoelectrodes to be above the single cell surface. We bring them very, very close. So I'm just going to break down here briefly. So we position this electrode to be 140 nanometer using scanning electrochemical microscopy. So this whole process of approaching can be monitored using we call them scanning electrochemical microscopy approach curve, which can further be simulated using COMSOL multiphysics to accurately determine the distance as well as the cellular permeability as well. And after that, we stimulate the neurons using chemical stimulation. Here is a high concentration potassium solution that will observe the dynamics of the release signal. So, you have also heard from Christy how powerful, right, amperometry to study exocytosis can be. So I want to point out, another, one of the observation we have here is when we have this nanoelectrodes to be very close from the cellular surface. So we actually were able to observe the exocytosis of acetylcholine with a high signal to noise ratio. Because we did a control experiment, basically the probe is pretty far away, several micrometer. We didn't do the nano positioning because it's kind of far. For that scenario, the signal to noise ratio of the measurement is pretty low. So that kind of makes sense. As the transmitter released through this exocytosis process, so they get diluted, right? So that's why when we have using SECM to position this probe to be pretty close from the cell surface, we were able to observe exocytosis with a high signal to noise ratio before it gets diluted. So this actually can have some implications for study diseases later on, especially at early stages of the diseases, if the changes are pretty small, right? But if we can, use this approach my group have developed, we were actually able to resolve some of the small differences at the early stages of the diseases. So besides single cell, another thing I didn't, I don't quite include data here because of time limitation, is we actually use this platform to unveil the mechanisms as well. So Christy shared with you about exocytosis. So looks like, in the literature, there's also another form that transmitter can be released from the cells that is directly released from the cytoplasma instead of packing the neurotransmitters into the particle-like vesicles releases them. That is exocytosis. So the criteria to identify that is to carry out calcium-dependent studies. So when Mark Whiteman came to visit UIUC, I had a very nice discussion with him, and so following our discussion, we carried out the calcium dependent study, and we learned that for this neuronal model, we are working with in collaboration with my colleague Professor Swider's group at UIUC, which is Abilene Californica. So this model actually follows exocytosis mechanism for the release of a neurotransmitter of cholinergic transmitter from the cell. So besides a single cell direction, another direction we also have been focusing on is the single synaptic study of neurotransmission. So synapses is the nano junction that forms between the presynaptic and postsynaptic neuron, and part of the brain communication occur through the synaptic neurotransmission path besides volume transmission. So because this nano gap is so small, so we actually need a nanoelectrode that can be positioned using scanning electrochemical microscopy to be in this nano space. So that's what my lab did. And after we position the nanoelectrode and the synaptic cleft, then we stimulate a neuron with high concentration potassium stimulating solution. And we observed diverse dynamics. So including a single peak and 2 peaks and multiple peaks. And we did quantitative analysis. So the average number of molecules we get from the double peak is twice of that of a single peak average. So this one is much more higher. So based on our data analysis, so then eventually, we propose a new mechanism that is the multi-vascular mechanism. So namely, the single peak here is correspond to release of content from a single vesicle. Then as the blue dot is the neurotransmitters. As the neurotransmitter gets released, so then you see the increase in current. So then it starts decrease because there's other processes going on. For the case of acetylcholine, it can be broken down by the enzyme. So a second contributing factor is the diffusion of the neurotransmitter, perhaps out the collapse, and also reuptake with the receptor is also can contribute to that as well. So for the two peaks here, so our hypothesis is before the first peak corresponding to the release of first vesicle decay to the baseline, there is additional input from the second vesicle V2 here. So that's why we have a second peak. Likewise, for the multiple peaks. Yeah, so overall, we're still in the process of a continue exploring this. For example, I have a collaborating project with two other groups at UIUC, where we are developing a glutamate sensing electrode to probe the glutamate neurotransmission at neuromuscular junction. So that was like a project, we get funded through NSF. Okay, so last thing I want to touch briefly here is besides getting information about neurotransmission dynamics, we can also get information about number of vesicles per area using electrochemistry. If you dig into the literature, the commonly used method to get this information is by doing cryo-EM. So here, we demonstrated that, using electrochemistry, we can get this information. So our data actually is pretty similar to the cryo-EM data here. So how we do that is, when we have a nanoelectrode of 750 to 860 nanometer, so we did analysis. What we are measuring here is multiple vascular content. So then we take this divided by the single vascular content, then we get a number of vesicles. So by doing this, I kind of shared with you all some of the new directions my group have been taking. So, near the last, I want just quickly to thank you, my group, as well as our collaborating groups and the funding support, from the agencies. Another thing I would like to mention is, we're also going to start a new direction that is to explore the gut-brain axis, which is also an emerging, area of research for the brain chemistry, as well. So with that, I'm going to stop here, and I really look forward to the discussion.
Okay, so May, you sparked a lot of questions with your CV curves. So I'm going to take, I was chatting with Jill and Janine, telling them we're going to take a little pause so that they can ask you the question, since I was asking them if they were going to go on to CVs or not.
Certainly.
So Joy, do you want to articulate your question?
Yeah, thanks, Jordy. So Jaime, that was super cool. I just wanted to ask about the shape of the CV curves and generally what governs the onset potential in the shape, because my understanding is that the current is driven basically like a battery, but the electrode is liquid where the ions are moving into the electrode. So I'm wondering, what kind of chemical, I think there was a question about chemical specificity, because I think any organic soluble ion could drive that current, but then also what potential that would be at and why it looks more like a reversible reaction rather than a capacitive reaction.
Right. So for the CV curve, so we actually, we are not observing anything new. That is pretty typical for any micro and nano electrodes. Even the electrode is like a nanocarbon or nano platinum. That's always the sigmoidal shape you can observe. So you can actually get the current potential curve. I actually teach that in my electrochemistry class, right? You can get the relationship for that. If you make a plot of that, there's two kinds of fractions, then it gives you a curve like that. So this is very different compared to the like macro electrode, like a large electrode. So another way to picture that is really the diffusion, right? So for the large electrode, you have like dark shape, right? You have a decrease after the peak current. So, for the curves we are observing, you get like plateau. We call that diffusion limiting region. So the difference for that is really the different diffusion profiles at the macro electrode, like millimeter, centimeter, in comparison to micro or nano electrode. So for the large electrode, like centimeter size electrode, you have a linear diffusion. And for the nano and micro electrode, you have like spherical diffusion. So that's why, for the large electrode, once you eventually your diffusion cannot catch up, you see a decrease in current. But for here, you actually can maintain like a diffusion, limiting region. Yeah, so those are the things, very good question. Hope I answered your question.
Yeah, thanks. Can I also just clarify for what governs the onset potential? Because I know in CVs, usually we're talking about a redox reaction. So that's like the redox potential. But in this case, you're not actually doing like an electrochemical reaction. You're doing more like a ion moving into a phase. So I'm just curious, like what governs that onset potential?
So I would say here, right? So what we are doing is an electrochemical reaction. So, the textbook I'm using, which is the Bart Faulkner Electrochemical Methods book, right? If I know like this, like ITIES, it was less common in the States. It is, this is the whole history of electrochemistry actually comes from the, mercury job electrodes, right? Plurography. So after that, that comes to the ITIES electrode. So two of the concepts I taught this spring semester for electrochemistry, one of them is you carry out electrochemical reaction based on the redox reaction. And the other one is, what I just presented based on the ion transfer and liquid ejection. So this is part of the reason I kind of shared with the deriving process of a Nernst equation. Yeah, so essentially it's a similar thing. The way how you picture that is just how you picture a redox reaction. I don't know if I answer. Yeah, so if we go back to, let me reshare my slides. Let me just slideshow. Sorry, everybody. Yeah, so this slides. So eventually, right, this is the theoretical basis. So if you derive from the chemical equilibrium for this ion transfer and liquid ejection, you actually have a Nernst equation. This is the same form as Nernst equation. So the way to picture this whole reaction process is just like how you carry out a redox process. So, sometimes, in the literature, when we talk about the pipette, right? So there are like PET probe. Those are different from this. PET probe, you basically have like aqueous solution inside and outside. But here, we actually have two different phases, inside and outside. That's why you actually form like liquid ejection. So here. Is that I answer your question?
I guess maybe I need to think about it a little more, but I don't want to take up too much
Joy,
time.
you want to think about this in the context of what you heard yesterday about conduction? When you asked the question about how is it that the anions are contributing to the conductivity, and you got the answer that you're building a concentration profile, so then diffusion and migration become opposing forces, right? So this is, you're building a potential because of a difference in concentration of the ions, and that creates a flow, right? The chemical potential is different. Not all electrochemical feedbacks need to be an electron transfer event. Ions can create their own electrochemistry as well. And this basically is the essence of why I love having this panel, because that's what comes up when you talk about concepts of bioelectrochemistry. There's a lot of talk about ion flow and potentials, right? Those create potentials as well. And so that's what that equation is basically telling you. If you take the Nernst equation and you change the concentration, you're going to build a potential, even if the species is the same. If its concentration is not the same, you have a difference in chemical potential, and that creates a redox potential or a membrane potential. Okay? So that's what the essence of what she's showing is. Does that help?
Yeah, I think that helps. I think I was just making an analogy to like pseudo-capacitors or capacitors, because that's also driven by Nernstian shifts due to Coulombic charges. And the shapes of those CVs are very different. So I think that's why I was getting caught. But
for different reasons. Supercat are still electron-driven processes, and these are ion-driven. The last ones that May talked about, they're ion-driven, but those still do electrochemistry. I know that we say electrochemistry and not, I guess, ionochemistry, but it's still, in the end, a current is just a flow of charge. Whether that charge is negative or positive is completely irrelevant. It's still a current. So that's how this kind of gets framed. And again, it's nice that you asked the question, because it connects very much to what you were asking yesterday. It's a similar idea, is that you're building gradients of ions, and that creates a potential. That creates forces that oppose each other.
Thank you, Joy. Very good question.
Yeah, so May, I put this on the chat to you. You always spark a lot of discussion with this ITIS. I remember this happened when you talked in person too. And it's because in electrochemistry, there's a lot of bias in favor of just thinking about everything as being electrons moving in and out. Right? Julie, you also had you were asking about the shape of the CVs as well. Did this answer your question before we move on? And I'll keep the other questions for after the rest of the statements.
Yeah, that answered my question. So thank you. I appreciate it.
All right, perfect. This is going exactly as I hoped it would. So we're going to redress and finish our statements. I have a list of those of you that have been posting questions. I'm going to reread the chat because it's exploded and I have lost track of it a little bit. But we're going to go to Jill Venton from the University of Virginia, and she's going to give her statement, and then we'll take it from there.
All right. Thanks, Jordy. It's a pleasure to be here today. And I'm just going to talk just a little bit about in vivo electrochemistry and just what I think the challenges are. I'm not going to show quite as much data. But to me, we've been using these carbon electrodes you've been hearing a lot about to try and unlock the secrets in the brain for a while. And again, this is a regular carbon fiber electrode here that you've been hearing about, and they've really been great, in a way that like these are stable and people have been using them for years. And carbon is kind of the secret sauce for neurotransmitters, that a lot of the amines do really good electron transfer to carbon, and they don't do it to like metal surfaces. So we want to know how to stick these in the brain, because the overall goal of a lot of the questions is to look at things like how do neurotransmitters change over time, and how does that kind of like neurochemical signaling control behavior. So I'm kind of pushing into kind of more of some of the in vivo things rather than looking at kind of cells in the dish. And so here are some of the challenges that I think are there. So I told you carbon is like the best material for these electrodes. And as you probably already know, there's tons of new kind of forms of carbon out there that you can now use instead of kind of normal carbon fiber electrodes. And so I think the field is kind of going through a process where people are trying to sort through which ones of these are really useful. I just put a couple pictures up, but there's, graphene, graphene oxide, carbon nanotubes, nanohorns, nanodiamond, nanoflowers, you name it, right? exists. And part of the problem is that oftentimes the research comes up a little short. It says that it's good, but we don't have a lot of comparisons in my mind. The other thing is that we need a lot more fundamental studies, because I think if we can really define what type of surface works for certain chemicals, what the properties are both of surface structure and surface chemistry, then you'll be able to, I think, take a critical eye and looking at new materials and sort of predicting whether they're going to be the material you want for a certain application or not. So our lab's been working a lot on actually surface structure. We like these kind of aligned nanotubes and nanotube yarns, but what we found is that they have little crevices in there. I don't know if you can kind of see, there's little holes in between them. And believe it or not, those little crevices are deep enough to cause a completely different type of electrochemistry than normal, and that's thin layer electrochemistry. And so I think that people have thought a lot in the past about chemical structure of the surface, putting oxide groups and stuff, and that's still important. But I think we also need to think about surface structure. So there's a lot of materials chemistry, and this comes up in tons of other fields too, right? That I think is really important for the neuroelectrochemistry field as well. If we're going to design better sensors, we've got to understand this surface interface. We've got to understand the materials that we're looking at. So that's one of the, I think, kind of hot topics. The other one that, again, I'm biased towards my things that my group are working on, but the problem with the carbon fiber, it's like good and bad. You start with something that's relatively rigid and you can make a couple shapes out of it and that's it. Right, but if you don't want those shapes, if you want to come up with something else, then you're not probably going to make it out of a carbon fiber. And so we've been working on a 3D printing process instead, which allows you to make whatever design you really want. And so in this, we take like wires and we put them in photoresist, and then we use a 3D printer that's built on 2-photon lithography. So basically you got to have 2 photons hit at the exact tiny little spot, and that polymerizes the photoresist. Anything that's not polymerized, you wash away, and you can end up with then photoresist electrodes. And anybody in the carbon electrode fields knows that you can then pyrolyze photoresist, right, to get carbon. And so that we can end up with pyrolyzed photoresist. And so these are some pictures of actual ones we've made. We've made spheres, we've made cones. And believe it or not, this is a nano electrode that we 3D printed. One of the things that happens when you pyrolyze is that it shrinks. And so we can actually make 3D printed electrodes that have a resolution smaller than the printer. And that's because it shrinks 5 times when we when we pyrolyze it. So I think that if you want to start thinking about like a completely different shape than what we've normally been doing, then being able to use some of these other kind of technologies are out there, emerging technologies for like tiny 3D printing, I think is going to be really fun and exciting. So I think there's still a lot of work going on kind of in electrode design, and there's tons of fundamental electrochemistry kind of going in there. I did want to mention a challenge I think that is underappreciated, and that is the data analysis challenge of how can I detect multiple neurochemicals in a mixture. Because the problem when you go in vivo, like Christy was saying, she could culture a specific type of cell and maybe it has 1, maybe 2 neurotransmitters. And then I go in the whole brain, I don't know what's there, kind of thing. And even when I can kind of predict, it's hard. So we often look at our data using these things called color plots. And so there's potential on the y-axis, time on the x-axis, and then the current that we see is in color. And I think we've underutilized them because I showed CVs up here. And, we're as electrochemists, we just love CVs, right? We love them so much, right? Like they're like the heart of like, cyclic voltammetry measurement, right? But if you think about it, you get a lot more data and kind of a picture of what things look like if you look at it like in this 3D manner, because for a lot of compounds, the CVs change over time. So this just happens to be a CV of adenosine that looks early, and we get 2 peaks where a primary peak is bigger, but over time, the secondary peak grows in. And so this doesn't play well with traditional data analysis techniques like principal components analysis. Like for dopamine, the CV rarely changes, but for other things, they change. And so we're pushing ahead, but I don't think we necessarily have hit it yet to try to use image analysis processing to look at the color plots as an image, like a microscope image, where we're borrowing their techniques. But there's a lot more like machine learning and data, big data, I think, that has just really not come to this field yet. There's been a few forays, maybe, and it's really hard because how do you calibrate from in vivo to like your beaker, right? Can you just run a CV in a beaker and then match that to something in vivo? You know, we all probably are like, no, but then if the answer is no, then how do you calibrate that? And so I think the data analysis, though, is huge. And if people can solve data analysis issues, I think that that's going to be really hot. All right, and then somewhat controversial for perhaps an electrochemistry talk, there are new competitors on the market that the neuroscientists are extremely excited about. And I think that I must say that every grant I write at the moment, I'm asked, why don't you just use these fluorescence techniques, right? Why don't you just use these fluorescence techniques? And so I think that it's like good. It's going to have to, we have to prove our mettle, right? You know, or people are going to be like, oh, good. Nobody needs to do that FSCV stuff anymore. It was too complicated and I never understood it to begin with. They love fluorescence in neuroscience. So here's they have these fluorescence based sensors, and I'm not going to explain them too much, but basically they're genetically encoded sensors. So they can get them to go into certain cells. And so this is what's called a grab sensor for dopamine. And what our lab has done is that we put our electrode, this is an electrode, in next to a cell that's expressing one of these fluorescent sensors. So actually, this is just a brain slice. This is in vivo. If this will work, will this work? Just a second. I got to go out of laser pointer mode, I think, to go back to the arrow. No, I didn't get out of laser pointer mode. Okay, now I don't know how to get back to a arrow. Okay, all right, well, anyways, but you can stim a cell and it'll change in fluorescence, and that'll tell you something about dopamine. So this is just one example. Oh, I see I didn't label it very well. The black trace here is our electrochemistry trace, and the blue trace is what you get with fluorescence. And so it's sort of interesting because I talked to one of the inventors of this and I said, you're slow. You're not really following the time course. And he said, of course we're slow. We make our sensors to be slow because the more it holds on to the analyte and doesn't let it go, the more it's going to give us fluorescent signal. And he's like, so the best sensitivity sensors are slow. And we all know that. Well, okay, he knows that. But I don't think anybody who picks up the technique probably knows that. So I think there are still some really good reasons to use electrochemistry, especially if you care about this downward trend, which is what we call uptake in neuroelectrochemistry land. But we're going to have to kind of prove that we can do things they can't do, which is kind of why I also think that data analysis is really critical. That if we continue to say, all I want to do is look at dopamine, people are going to say, yeah, thanks, I'll just get this. And it's going to be harder for us to kind of compete if they can do this. And in all honesty, if you know it's slow and you have a competing technique, you can deconvolute it. So we can deconvolute the fluorescent signals if we need to, right? And then you could just go use those. So other challenges I didn't make slides on, but we could talk about. Behavior is extremely challenging, right? To get animals to do things with electrodes in their head you know, causing noise and that kind of thing. I really think the field is going, as I said, multi-analyte and probably towards arrays. Again, there's been a fair amount of people who worked on them and nobody who's really conquered it. That's my assessment of the field. People are working on smaller model organisms. My lab works on Drosophila. Other people work on things like zebrafish. These are really great for genetics and biology, but they're small and it requires a lot of things. If you want to hear about, we actually can really now do in vivo behavior in a Drosophila, which I thought was probably impossible. So like you can get there, but it's hard. And then the last thing is whether these things should ever go to humans. Well, let's just, and people are using it a little bit. When they do, it's typically in some human that's already having brain surgery for something else, right? We don't just stick in electrodes in people who have no issues, is there something there that can inform people? And so I think that these are kind of interesting challenges to think about, and ethics and that kind of thing. All right, so with that.
Thank you, Jill. We're going to run to our last opening statement, and then we'll continue our discussion. So our last panelist, and not least for sure, she's been super active in the chat, is Janine Mauzeroll. Hopefully I pronounced your last name correctly. From McMaster University. Oh, McMaster or McGill? Oh no, is it McMaster?
No.
McGill. Okay, I get the two constantly mixed up. Sorry, my apologies. McGill University in Canada.
No,
Go ahead, Janine.
Somewhere up north, right?
Sorry, I'm a little distracted with all this chat is really blowing up, and I'm
No
having some
problem.
trouble keeping up. Go ahead.
Thank you very much, Jordi, for the nice sharing and encouraging people. It's been really great, and I enjoyed seeing all the wonderful things that Christy and Jill and May have been doing. I'm Janine. I'm in Montreal, and I was trained as an electrochemist in the BART group in my graduate work, and then I did my postdoctoral work with Jean-Michel Savéan, who's a theoretical chemist. So I guess what I want to say is that A, I have a French accent, and B, everything that I do in electrochemistry is experimental and tied to theory, because a lot of the times the equations that we learn in our undergraduate level classes don't actually apply to anything that we're doing in our graduate work. So they posed us the question, what is the future of electrochemistry within the context of neuroscience application? And I guess in my group, we work a lot on detecting non-electroactive species, namely D amino acids in the brain across single cells, primary cells, rat, mouse, and human, and also in vivo in the brain of Xenopus laevis tadpole. And so that's our main focus. How can we detect non-electroactive species within the brain? And our strategy is one that Christy mentioned in terms of modifying some of the nanoelectrodes and microelectrodes with enzymes. And then, this opens up a whole can of worms in terms of, well, what happens to your enzyme activity? And yes, you might be generating some hydrogen peroxide, which is electroactive, which you can detect, but you're not going to have 100% capture efficiency. So how do you deal with the quantitative aspects of this? And for us, that's always tied to finite element numerical simulation of the entire process. So we detect non-electroactive species, we carry out simulations, and then in the end, I think our greatest challenge is actually training people in all of the fields that are required to do great work in neuroelectrochemistry. I mean, most people have had something like half of a class in electrochemistry when they get to graduate school. And so they must learn this. But in neuroscience, they must also learn how to choose an instrument, for example, in order to get the best signal to noise. That requires some knowledge. Then they're faced with the data that Jill was showcasing. I mean, that's a lot of data. And you're going to be reproducing this quite a bit. So they also have to learn how to handle this data, learn to code, learn to develop maybe algorithms that will make their work efficient. That's not always a skill that chemists are equipped with, And I guess if I had a critique of our field currently, is that there's not a lot of sharing, And a student will invest a great deal of time in developing these algorithms. Sometimes they'll do it in MATLAB, but that requires a license. And so when they move to a postdoc, in a lab that maybe doesn't have this license, then they kind of lose this knowledge, right? So I really do think that promoting data treatment on open source platforms and then making these available, like for example on GitHub, such that other people can easily modify this and learn, this is going to be very helpful to the field of neuroelectrochemistry. It's also conducive to better graduate student training. And so one of the things that we find really hard in terms of moving across cell types is that you really need to have reproducible sensor fabrication. I mean, I think Jill's state-of-the-art sensors, like you can definitely have a quality control there to know, okay, does it have the right geometry? But in neuroelectrochemistry, it still requires like an enormous amount of controls in terms of calibration curves before and after, especially if you're making the measurement inside the brain, right? Because you cannot control the entire matrix. Sometimes it's an excised brain. Sometimes the insertion protocol may be quite complicated and you may damage one of the layers that you thought were going to mechanically protect your sensor, at least for us with the enzymes, you know? And so it's important to be very rigorous from an analytical chemistry point of view and carry out calibration curves before and after your measurements in order to get reproducible sensors. Also, there are very few sensors that are actually commercially available, which limits, in my opinion, like the widespread use of some of the beautiful sensors that you see in the papers. You read them, you think they're wonderful, but then, you know, you would have to invest the time of someone for at least 18 months, you know, to get this type of sensor along. So in the same way that we should be sharing as a community much more our data treatment algorithm, we should also be sharing much more our knowledge about probe fabrication. I think it's extremely beneficial for graduate students to be able to go abroad in international internships. You know, you can learn how to do a probe in a lab more rapidly than if you were doing it yourself in your lab. I don't know in the US, but in Canada, we have several funding ways by which people can go spend like 6 or 8 months in a lab. Like my student that was working on the enzyme neurosensor, she spent 6 months in Italy learning how to make mutants because I don't know how to make mutants, but in the end, it was overall beneficial. The other thing that's a challenge in neuroelectrochemistry is corroborating the electrochemistry with an alternate method, which I think Jill talked about in terms of the fluorescence. You could try to corroborate there. There are also HPLC methods as long as the time scale of it permits it. And for the behavioral animal studies that we do, it's a real challenge to corroborate the electrochemistry to, like, for example, animal movement. You know, like you basically carry out your experiment, you monitor with a camera, and then you have to track the motion of all the Xenopus laevis tadpole, which are basically baby frogs swimming around. You have to track their motion and quantify it, again, with image processing and large data. So I think one thing we should do in the future is incorporate more of the aspects that we're asking our students to learn independently, namely, you know, large data treatment. We should welcome this and talk about it and find ways to make it more efficient. And the second thing is, I think we should always promote collaboration if it allows the students to reach their goal quicker. So from my view, the hardest thing in neuroelectrochemistry is equipping our students such that they can become great scientists, because without all of these items, I don't think the field is going to move forward. Thank you.
All right, Janine, thank you very much. So we've had a really active chat, and we have a list of people that have been posing questions. I'm going to start going through that queue so that we can have them ask their questions live. The first one that I have in my queue is Wan-Yu. Do you want to restate your question? You had a follow-up at one point because you've been exchanging information with Janine. Do you want to ask where you are now?
Yeah, sorry, I have a lot of questions lined up, just pop out.
That's what we're here for. So go ahead and ask.
Yeah, so all my questions are kind of related. So my background is AFM, but not particularly SECM and SICM. Well, my background is actually supercapacitor. So it's a specific type of electrochemistry. So I think I was asking of the impurity because I was using AFM to look at the double layer structure on the electrode for supercapacitor purpose, not bio purpose. So I noticed what was in the chat about the reproducibility, because measuring in liquid in general is kind of challenging, and that's kind of tied together of why I was asking about the resolution, special resolution, and also the downsizing part is all linked together. So just now you were mentioning how do we start from one single cell to go to the ensemble, but for our case in terms of device, it's we know the bulk performance and then we downsize it to the nanoscale. It's the other way. So I always, when I do the measurement, I always have in mind is this curve looks different than the electrochemical curve looks different from the bulk that I know very well. So is this a real signal, or is there impurity in the system? What are the special resolution I can count on? So let's say your nano probe is, I don't know, 1 micrometer, but you have all those diffusion going in that your total size is probably including a lot of grains. So how do I know, for example, if I imagine I have a substrate have multi-grain and my probe size is just on 1 grain, but you have to take into that all account. How do I know what I'm probing? That's kind of why all my questions are all tied together. Yeah, so I don't know if you have a comment on this.
The way that I would elevate that is single cell, really single cell. Or is single grain really single grain? It's really the same question in a sense. So any thoughts on that?
Yeah, so I guess I would tell you, Wang-Yu, that it's possible to know if it makes sense by using numerical methods. You could, like finite element method, it's a method by which you can mesh your entire electrochemical domain, and you can play around with the geometry of your probe. And then you can simulate, because let's say in an AFM tip, you'll have a diffusion profile that's very different from a beveled carbon fiber, right? So using this tool, you can simulate CVs in the bulk and then define an area where you have your surface. This will take into account everything that if you set up the boundary conditions correctly, everything that has to do with the hindered diffusion, the change in kinetics, you can have a lot of control over it. And it's really pretty straightforward to start working with this. There's no unique equation, or there's no way you can focus in on a single parameter. It's a multivariate type of problem. So in my view, it requires simulation.
Any other thoughts on that along those lines? All right, Wang-Yu, did that answer your question? Any follow-ups? Because you had a few questions, so I wanted to make sure that.
Yeah, I think so. Yeah, they're all kind of tied together.
Well, there's a general theme that is emerging in the different panels of this idea of you're measuring control systems, and what does that really mean for the system? In what they call in vivo. If you're making a device yesterday, it's more like the industrial level system, right? And I think what you're hearing is that that's still an open question. Udari, I have you next in the queue. Are you able to ask your question, or should I just read?
Yes, today I'm good.
Okay, sounds good. So go ahead and ask your question.
Thank you, Dr. Jati. Actually, I would like to know, as we all know that ions play a role for especially double layer formation. And also when we talk about that centimeter scale electrodes and MEA or any electrode device, that cations and anions sometimes promote the reaction, sometimes deactivate the catalyst by depositing like carbonate or hydroxides. So such kind of things. So I would like to know how these ions play a role in this type of systems. And also my second question is basically the reproducibility and stability of these systems, especially biological systems. These are really sensitive. So how do you do they check their reproducibility, and how those are stable? So that's all my questions.
All right, who wants to take it on?
I can say something about the reproducibility. Unless May, you were ready to go, you're fine.
Oh, go ahead, Christy.
Oh,
Please.
I'll chat a little bit. I'm sure May will have something to add. You know, the reproducibility piece is something that, you know, my group definitely struggles with. I mean, we essentially just end up setting benchmarks for, you know, what noise is acceptable on like a background current, right? When our electrodes are in solution. And you kind of just get a sense for what is the range. I mean, we use the same kind of signal to noise considerations that you use all throughout analytical chemistry, right? In terms of what we can detect and not detect. But, you know, as I mentioned in my little bit of intro, I mean, fouling and increase in noise, like as you make a series of measurements is definitely a problem. And, you know, Jill maybe can comment on this even more than I can, but you know, when people are doing in vivo measurements, my sense is that they, you know, essentially do a calibration measurement at the beginning of using an electrode and at the end of the experiment, and kind of try and normalize based on how the electrode performance changed throughout the complex, like the use of the complex environment. There's no kind of perfect answer to avoid issues with reproducibility or fouling, but just kind of correction factors to keep in mind is how my group handles it.
Okay. Yeah.
May, did you want to add your thoughts now?
Yes, I also want to mention briefly for the like nanoelectrodes. So, you know, it's really like emerging area of research, and we keep discovering like new phenomena. So, you know, so for example, you know, all of these new phenomena, we have like, you know, counter strategy, right, to make them even better. So for example, another thing I didn't quite share is, you know, while the work, while I was still postdoc in the Amamiya lab was, you know, where we observed the nanoscale damage to like a metal electrode just by like static charge from a person. So those work actually eventually adopted by the Harris quantitative chemical analysis textbook. So basically, you know, the static charge from a person, you know, wouldn't do much to like a large electrode. But for like nano carbon platinum, it can damage the electrode surface. So, you know, likewise, you know, we came up with strategies that is, you know, for example, now my students handle platinum carbon nano electrode in the lab. They have to wear this like protective gear, right, to grounding themselves, things like that. When you do that, you know, so you actually get a pretty good nano carbon or platinum electrodes. So the point I just want to make here, right? Of course, there's lots of things, you know, we still need to learn, but, you know, we are really learning more and more about, you know, the phenomena at this emerging nano science. So.
Just a follow-up to that, I find that crazy that you have static poison in your. But this, I mean, you know, we're going to have a panel on measurement of local electrochemistry, and I feel like this is going to come up again, more from the perspective of the methods themselves rather than the application. But one thing that came up yesterday when talking about issues of reproducibility and benchmarks was, you know, are there in this domain reference publications that a student could go and read and say, you know, if you want to know that you're doing this correctly, you have to follow this kind of like checklist to make sure that you're relevant. Does that exist?
Yeah, I can put the paper in the chat. So
okay.
It might take me a couple of minutes to find it. It is a paper published in analytical chemistry. And we also have the strategies, you know, what precautions you can take when you work with metal or carbon nanoelectrodes. So
Right. And so what I would say, broadly speaking, is that, it doesn't matter what you're detecting. Fouling is going to be an issue with any system as long as you're using a nanoelectrode. So if you can put that paper there, I think that there's plenty of people that are going to want to read it.
So I'm not talking about the fouling here, Jordi,
Right.
So I'm talking about electrostatic damage to the surface of the nanoelectrode.
Right, but this is something that happens even before you do the measurement, right? So it's not. In other words, it's not a
process.
But it's not an issue of you measuring biological samples. It's going to happen as long as you want to use a nanoelectrode.
Yes,
correct?
This is general to bio, non-bio. So
right. Yeah, so that makes it very attractive. The issue of methods papers has come up, and the students sometimes, I could sense that they're a little confused in terms of like, well, how do you, reproducibility is important, but how do you go about it? And so, yeah, if you can share that paper, that would be a very good example of what these methods mean.
Yeah, that's right.
All right. In the interest of keeping the discussion going, because we're running out of time, Aditya, do you want to go ahead and ask your question?
Sure. Thank you so much. I think, chat was really helpful in understanding what I had. I had a question about the double layer at the nanoelectrodes, but I guess that was all partially answered during the discussion in chat. So I have a little bit of like a bigger picture. Hi, can you hear me?
Yes, you broke off for a second, but you're fine.
Okay, sorry. Yeah. Okay. Yeah. So I wanted to say I have some like sort of like a bigger picture question, because I'm coming from a lab where I am my advisor's very first student. So I had to build and set up the lab, and it takes time to get the actual research done while I was setting things up. So my question is, what is the best way to have the information transferred? I think that was discussed in the very last segment of the opening statements. And because similarly working labs may not tend to share everything because I think they might fear being scooped up, or it could be like they would want more citations because there's an ever increasing competition to get more and more recognition for the work. So how do you think as in your position, you can influence a shift in attitude so that we don't prioritize this too much to boost these metrics, so that we can have like a faster science done, or like more easier information transfer?
I'll start. I think that one of the keys is to do your best to kind of meet people, because like cold emailing somebody is tough, right? I don't know who this person is. Should I invest my time in them, et cetera, et cetera. But when somebody, we've had a conference discussion, we've met here, et cetera, et cetera, you can point back to something we've done. So this is something where, like, even if you're not ready to present at a conference, you might try and convince your advisor to let you go, just to make those connections. Because a connection with a group or a person that I've met before, I'm a lot more likely to probably open up and say, hey, come down and do that. But we go all the time. I mean, like, it's not, I sent somebody down to learn fruit fly in vivo techniques, we went brain slicing techniques, et cetera, et cetera. So I really think that you're right, that it's tough. But the other thing is just to realize that you're right, it's not all written down. And that's a tough one, when students kind of realize, oh my gosh, not all the steps that I need are in this paper, crud. So, that's a thing for trying to get to talk to people one-on-one, if you can.
Maybe I can add something to that, because of course, not every group, especially new groups, necessarily have money to send people to conferences to present. And it I don't know, it's unfortunate if that's a barrier. A lot of conferences definitely have travel grants, especially if you have like financial need. And so even if they don't really list travel grant, write to the chair of the session, ask if there's money to help get you there. And I'll say, even if you can't get there in person, another tip, honestly, is don't email the faculty member, email the first author graduate student on the paper and talk to them first. Usually they'll consult with their graduate advisor before sharing information, of course, but, they are less busy and more in the gory details of the work, right? And now we've all learned a lot during COVID about how to do research when we're virtual. So, now we all have these cams that we can wear on our chest while we do an experiment, so someone can watch. So you can actually get, a virtual visual of how to make an electrode or ground a system or whatever it is that you're looking at. I mean, we all have kind of been forced to get that in place. So I think, that's a potential upside to what COVID has done for all of us.
Yeah, but I do think you shouldn't be shy, and you should play the violin. Everybody remembers what it feels like to start their lab. And so if you started off your email by, "Hi, I'm the first graduate student of blah, and my goal is to do this," I mean, I will answer you because I remember what it was like when I was trying to get my Puller parameters for my first nanoelectrodes, So persistence goes a long way, and play to what your situation is about. If you make it more humane, and people can relate to you rapidly, like don't write a large email, you have better chance of getting a response.
Yeah. Okay. Thank you.
Okay, one quick question to the panelists. We have 3 minutes left, and I know we have more questions than minutes we have left. Is it okay with you? Do you have to all run exactly at 11:30?
I have to run another meeting at 11:30.
Okay,
so I only have a couple minutes because I have children to pick up from camp.
Okay, just making sure that I couldn't abuse your generosity. Okay, so let me get through questions, and what I'll do is that I'll close it at 11:30, but I'll close it officially, but then I'll stay open and try to address some of these questions that may be left. Alessandra, you want to ask your question?
Hello, yes, thank you. Thank you for all of your talks. They've been absolutely wonderful. I have a pretty quick question, actually. We had discussed a lot about carbon fiber as the sort of gold standard electrode material, but there was a little bit of conversation about new electrode materials. What characteristics are you looking for that ideal new electrode material that would make you switch from carbon fiber?
Yeah, so I'll take this first. Briefly, we're looking for kind of electron transfer to see if we can get better electron transfer. You want to characterize that in all the kind of normal ways. We do a lot of impedance now to characterize that. And we're also kind of sometimes looking for, it sounds weird because we haven't really done it fundamentally, but things that might enhance certain reactions, and as I said, we'd like to figure that out fundamentally, but to kind of play off for selectivity. And then sometimes we're looking for things of different shapes that we often like surface roughness, because that gives you just more surface area. We don't want a large electrode, right? But we want high surface area. So things that give you surface roughness is often what we're looking at as well. And sometimes coatings that will help with things like fouling. So I'll keep it short.
Okay, we have 1 minute. Erin, give it a shot and see if we can get this done.
Awesome. So I think my question was mainly on reproducibility, which we handled really well in the discussion, talking about the importance of calibration curves, collaboration. And so I guess just from the biological side, because that's a lot harder to control, do you all have any advice for specific controls you should be including to kind of control for the different electrochemistry across bacterial species, across bacterial conditions among labs?
Yeah, so I guess across labs, that's really hard, but I think you want to control for time of exposure outside of the incubator, CO2 levels, pH, temperature changes, you have to have done these controls. Also, you need to test if you reduce your media to something more simple, you need to do all these controls under these conditions. If you add a molecule that you're going to be detecting, you need to evaluate the impact. So it's a huge to-do list, and you have to do this for every single cell line that you test. You can't assume that if you have a mutant, these controls are going to apply. You have to test it out every single time.
Okay, let me interfere. And if there are any other thoughts, we can keep discussing, but I want to be very respectful of the people that have to go and move on. So I'm going to officially close the panel here, and we'll stop the live stream. Thank you, May, Jill, Janine, and Christy for being here, for being so generous with your time and sharing your ideas. This was a really engaging discussion. So we're gonna close it here, but if people want to stay and discuss for 5 more minutes, we're gonna leave the call open in case anybody wants to keep talking before we officially close it until 1 p.m. So officially we're stopping the panel, we're closing the live stream, and we'll start again at 1 p.m. with local electrochemistry.